everyone, Ziausen this side. So today hand we are starting with a topic called as plant growth and development. Okay, so let's get started. So starting with the first thing first, here development is the sum of two processes called as growth and differentiation. Now, dealing with the first thing first, that is called as growth. So here, growth is the permanent or irreversible increase in the dry weight, size, mass or volume of cell. Or rather, I'll be giving you a proper definition of the growth, that it is irreversible increase in dry mass, and number of cells. That is a proper definition. Number of cell and the dry mass. Now, in plants, growth is accomplished by the cell division, increase in the cell number and the cell enlargement. So the cell will be dividing from 1, 2, 4, 8, 16 and so on. And the cell also enlarges as well. So, growth is a quantitative phenomena which can be measured with respect to time. Okay? Deal with. Now, plant growth is generally indeterminate. Now, what is the meaning of this? So, basically, due to capacity of unlimited growth throughout the life, the meristematic tissues present at the certain locality of the plant body are continuously dividing and redividing. Okay? Deal with. Now, what does this meristem tissues means? So, basically, they are the specialized cells which have the capability of division and redivision and can form any type of the organ. Okay? Now, the plant growth in which new cells are always being added to the plant body is due to the meristem activity. Okay? And do remember one thing, that in special plant in which the meristem are continuously working, they are called as open form of plant growth. Now, the root apical meristem and the shoot apical meristem, we call them as ram and sam. Okay? The ram will be root apical meristem, whereas the sam will be shoot apical meristem. They are responsible for primary growth and the elongation of the plant body along the axis. There is intercalary meristem as well, which is located at the nodes which produces buds and new branches in the plants. I repeat, ram and the sam are the apex or the extremities, whereas the intercalary meristem is present at here. The meristem are present at the nodes and the new branches in the plants. At the nodes and the new branches in the plants. Okay? Deal with. Now, the secondary growth in the plant is the function of lateral meristem, that is vascular cambium and the cork cambium. Okay? The secondary growth is the plant in the secondary growth in the plant is a function of lateral meristem, that is vascular cambium and the cork cambium. Now, basically the secondary growth is the increase in the girth, whereas the primary growth is the increase in the height. This will be the primary growth, whereas this will be the secondary growth. Now, the phases of growth. So here, the formative phase of enlargement and the phase of maturation. So the first phase is called as the formative phase, in which it is also called as a phase of the cell formation or cell division, which occurs at the root apex and the shoot apex, which I will be calling it as ram and the sam. Like the ram and the sam function. Okay? Deal with. Which have the meristematic tissue. And the rate of the respiration is very high in the cells which are undergoing mitosis during in the formative phase. So, continuously division and the redivision is occurring in the formative phase at the meristematic tissues, whereas the phase of enlargement, the second phase, is the newly formed cells which are formed in the formative phase. They are undergoing enlargement. The enlarging the cell also develops the vacuole that further increases the volume of the cell. The cell enlargement occurs in all directions with maximum elongation in the conducting tissue and the fibers. With maximum elongation in the conductive tissue and the fibers. Deal with. Now, dealing in the phase of maturation, what happens here, that the enlarged cells develop into special or particular type of the cells by undergoing structural and physiological differentiation. So this was the final phase of the growth. Like the first one will be the proper growth and the second will be the differentiation. Now, Going for the growth rate. So here we have two different types of growth. We'll be going through both of them. So increase in the growth per unit time is called as growth rate. Okay. Increase in the growth per unit time is called as growth rate per unit time. But it may be arithmetic or it may be geometrical. So if I will be dealing with the arithmetic growth, it is the rate of the growth is constant and increase in the growth occurs in arithmetic process somehow. Like let's say for example 1, 2, rather I should say 3, 4, 5, 6 and so on. Okay, deal with this will be arithmetic, and the formula for the calculation of the arithmetic growth will be LT is equal to L0 plus RT. Basically the arithmetic root is found in the root and the shoot elongation. Here the LT stands for the length after time, whereas the L0 is the length at the beginning, the initial length, and RT is the growth rate into time. R is the growth rate, whereas the T is time. So here we will be having graph in this fashion. Okay, continuously it will be growing. This is what we call as the arithmetic growth. But if you will be dealing with the second one, that is called as the geometric growth, here the initial growth is very slow, but it increases rapidly thereafter. And every cell divides here. The daughter cells grow and divide to the granddaughter cells that result into exponential growth. The geometric growth is common in unicellular organisms which grow in the nutrient-rich medium. Here, I will be giving you the examples like 1, 2, 4, 4 to 8, 8, 16, 32, 64, 128, 2, 56, 512, 1024, 2048, 4096 and 8192. Done. This 8192 is the 13th division of the one single cell. So sum of one single cell has given almost 8192 cells in its 13th division. This is what the power of the geometric or the exponential growth is. And the opposite one is called as arithmetic or logistic. So here we have the geometric growth in this fashion, where we have lag phase, log phase and the stationary phase. This one will be called as lag phase. This exponential phase is also called as log phase, and the last phase is called as a stationary phase. This is also called a sigmoidal curve. Okay, deal with. Sigmoid growth curve consists of fast dividing exponential phase and secondary phase. It is typical of most of the living organism in their natural environment. And here we have a formula as well, that is the W1 is equal to W0 e raised to RT, whereas W1 stands for final size, and the W0 was the initial size. R is the growth rate and T is the time of growth to the base E, and base E of the natural log, that is 2.71828. Okay, deal with. So the cells produced by the apical meristem become specialized to perform the specific function, and this act of maturation is called as differentiation. So one cell has been carried forward, it has taken its first step, and it is going to be a special cell. So this proper phenomenon will be called as differentiation, whereas the living differentiated cells that have lost the ability of the division, they can regain the capacity of the division, and this phenomena is called as de-differentiation. This phenomena is called as de-differentiation. Again, that is coming back and taking the ability of division, and the redivision will be called as de-differentiation, in which we can observe this in the interfascicular cambium and the cork cambium. Whereas the de-differentiated mature cells lose the capacity of the cell division and again perform for a specific function. This will be called as redifferentiation. So we have three phases. Differentiation, one single cell is going for a perform function. Then that after performing that function, the cell is coming back to the original position, will be called as de-differentiation. And again it's going for the performing the same function or the different function. It will be called as redifferentiation. Okay? Deal. Now, the sequence of the development process in the plant cell. So here, the different structures develop in different phases of the growth as well as response to the environment. And the ability to change under the influence of internal or external stimuli is called as plasticity. Please do remember this. Okay? Heterophyly is the example of plasticity, in which there is a subject of plant. If you are growing that plant into water, that plant will be showing different arrangement of the different type of the leaf, And rather if you are growing it on the soil, you will be having different type of the leaves. That is what we call as heterophily. Now, we have some plant growth regulators here, which are simple molecules of diverse chemical composition, which may be indole compounds, adenine derivatives, or the derivatives of carotenoids. Basically, like the hormones name will be the auxin, gibberellin, cytokinins, ethylene, and the abscisic acid. So the auxin was first isolated by the F.W. Wendt from the tips of coleoptile of oat seedling. Okay? And the second one is called as the Bakane disease of the rice seedling, that is the foolish rice. It is a fungal pathogen called as Gibberella fugicori, and the scientist name was Kurosawa. He found that this disease is caused due to the presence of the gibberellin hormone. Skoog and the Miller, they identified the crystal form of the cytokinin, promoting the active substance called as kinetin. So starting the first thing first, the first plant hormone ever. So here it is commonly called as indole 3 acidic acid, or what we call it as IAA, indole acetic acid. It is generally produced at the stem and the root apex, and the function is it is responsible for the cell enlargement and the cell division. It is responsible for the apical dominance. Please do remember, like if this is the plant, the auxin will be working in this fashion. Okay, it is responsible for the apex elongation. Okay, it is responsible for the inhibition of the abscission as well, that is aging of the plant. It inhibits the aging of the plant, and it induces the parthenocarpy, the ability of the plant to develop a fruit without fertilization. That is what we call as parthenocarpy. Without seed formation, rather, I will be going for. Gibberellin. The most common form of gibberellin is GA3, that is what we call as the gibberellic acid. GA3 is the most advanced one, or most best one, better one. Rather, GA1 is a kind of negative hormone. Okay, deal with. There are more than 100 variants of gibberellin are present. Now, here, cell elongation, breaking of dormancy, early maturity, and the seed germination are the function of gibberellin. Gibberellin is responsible for increasing the height of the plant as well, and the size of the fruit as well. Cytokinin, most common forms include kinetin, zeatin. They are mainly synthesized in the root, which means I'll be calling it as RAM. Their function is cell division and the differentiation, whereas they are responsible for they overcome the apical dominance, which means they are responsible for the lateral dominance. Okay, lateral dominance of this. Deal with. They promote the nutrient metabolism. Now, dealing with the ethylene, the gaseous hormone. Ethylene is the gaseous hormone which stimulates the transverse or isodiametric growth, but it retards the longitudinal growth. Okay, this is also responsible for the increasing of the girth of the plant. It is responsible for inhibition of the longitudinal growth. It is used for the fruit ripening. Yes, ethylene is one of the most important hormone for the fruit ripening. It is responsible for the senescence as well, and it is for the lateral dominance. Here like apical is written, but still it is responsible for the lateral dominance. Now, the abscisic acid. So abscisic acid is also called as the stress hormone, or also called as the dormin hormone. It is mainly produced by the chloroplast of leaf. The function is bud dormancy, leaf senescence, parthenocarpy, and the seed development, the maturation. The case is, if the unfavorable condition is present, the abscisic acid will not allow seed to germinate because of the unfavorable condition, that the plant may die. And after the favorable condition comes, the gibberellin will be acting now, and it will be allowing the seed to germinate. Now, a short topic that is called as photoperiodism. It is the effect of the photoperiodism, or the duration of the light hours, on the growth and the development of the plant. Especially the flowering is called as photoperiodism. So the amount of the light which is required for the plant for flowering purpose is called as photoperiodism. On the basis of the photoperiodic response to the flowering plant, to have been divided into following criteria, that is called as SDP, LDP, and DNP. SDP are called as short day plant, which require less than 12 hours of light, and rather, in other words, more than 12 hours of darkness without interruption. Even if a small flash of light is given to them in the dark period, they will not flower, they will not produce flower. Okay? The long day plants are also called as LDP. they require more than 12 hours of light. And the day neutral plant are the special types of the plant which do not require the proper more than or less than. They require a specific time of the light and they will flower. So ultimately, like the day neutral plants are flowering the whole round of the year, whereas the short day plant and the long day plant are seasonal ones. Okay, vernalization. It is a process of the shortening of the juvenile or vegetative phase and hastening the flower by a previous cold treatment. Do remember, vernalization is carried out by a hypothetical hormone called as vernalin. It is a cold treatment of the plant. So ultimately, the plant will flower now. So hopefully, this lecture has been beneficial for you guys. Thank you very much for your so much support. If you do have any kind of questions as well, you can ask me there in the comment sections. Okay, thank you.